Welcome to EduRev.
Welcome students. I'm Clarice Pereira Colasso and I have been training NEET medical aspirants in botany and zoology as well as NCERT based board exams for the past 8 years. In today's video, we are going to continue with our study on sexual reproduction in flowering plants. In the first video in sexual reproduction in flowering plants, we focused a lot on the pre-fertilization structures and events regarding the male reproductive system. In today's class, we are going to focus on the pre-fertilization structures and events, mainly speaking the structures of the female part. In this, we will study the structure of the gynoecium and we will study the structure of the megasporangium, which is nothing but the ovule, and the embryo sac that it contains. So to start off with the pre-fertilization structures, okay, we will start with the structure of the gynoecium. So the female reproductive part is called as the gynoecium, or alternatively you can call it as the pistil. Now the pistil or the gynoecium technically is made up of three parts. The uppermost portion is the stigma, that is the projected portion, the style which helps in the projection, so it's an elongated structure, and finally at the base you have the ovary which contains the ovules. Okay, so that is the major part of the gynoecium there. Now if you have a look at the pistil here, on top you can see the presence of the stigma. It is a receptive portion, a sticky portion. This is the part that receives the pollen grain during pollination. Then you have the style there, and the style helps in pollen tube formation rather, okay, and therefore it helps in the transport of the two male gametes as we studied in the previous video. And right down you have the presence of the ovary containing the ovules. The ovules technically form the seeds, whereas the ovary forms the fruit. Okay, so you have an outline here of the pistil. It's three parts: the stigma, which is a sticky receptive end, the style, which is meant for the transport of the male gametes by the formation of the pollen tube, and finally the ovary, which forms or rather contains the ovules that give rise to the seed, and the ovary which technically will form the fruit. Now you have a variety of gynoeciums basically depending on the number of pistils that it contains or the number of carpels that it contains. Now we can divide it into two: monocarpellary, indicating the presence of just one carpel or rather one pistil, and polycarpellary or multicarpellary, indicating the presence of multiple pistils, many pistils. Okay, now when we talk about many pistils, it could occur in two possible ways. For example, the pistils could be fused together, for which we will call the gynoecium as syncarpus, okay, or it can be unfused, they can be free, okay. In that way you will call it as apocarpus. Now the word syncarpus, syn implies synchronized or fused together, okay, whereas apocarpus, the letter A very clearly tells you that something is disjoint, something is missing over here, okay, so implying that the carpels are separated. Now let's look at figures of each of these types of gynoecium, starting with monocarpellary, a single carpel. So when we studied the previous figure of the pistil, a single stigma, style and ovary, we were talking about monocarpellary gynoecium. So this is a very simple form of the gynoecium. But let's take another example. Let's talk about polycarpellary gynoeciums. Okay, so this is polycarpellary with syncarpus. Syncarpus would imply that all the carpels or all the pistils must be fused. So you can see quite a big stigma over there, very fused. The ovary seems to be really bulged and really swollen because you have many basically many smaller ovaries that have fused together, many carpels that have fused together. So this is a syncarpus ovary, so big bulk structure. But if you look at the opposite structure, that is apocarpus ovary, okay, so apocarpus structures imply that they are not fused. You can clearly see that all the ovaries seem to be separated, the pistils or the carpels seem to be separated, so you've got all these smaller structures, okay. So we need to know the difference between the different types of gynoeciums based on the number of carpels or based on the number of pistils. Now if you take a cross section of an ovary, this is technically what you would see. Now depending upon the species, you will have different types of cross sections, different types of structures, different placentations, if you will. If you remember your 11th standard syllabus, you have studied about placentation. But here you have a technical a very typical ovary. Now let's study what are the different parts of this typical ovary. Okay, the first thing that you will find in the ovary is the presence of a number of ovules. So you can see that these ovules may be around 6 of them, these small bulk kind of structure. Okay, now these ovules seem to be attached, or they have a point of attachment, and that point of attachment is nothing but the placenta. Okay, so it starts drawing up nutrition from this particular placenta. But this placenta is not restricted to only the center, as you can see. The placenta can be found in two other regions. For example, the placenta can be formed forming the entire inner part or the inner circumference of the ovary. Okay, but this particular placenta also seems to be forming septa in between. In other words, it helps in the division of the ovary into smaller compartments. Okay, so placenta can be present in three different places. Now, suppose if you have septa, or rather placenta forming the septum, okay, so they help in the formation of these number of chambers. Now here very clearly you have three chambers. A chamber in a plant ovule is called as a locule. Okay, so we can say that this is a triloculated structure, the presence of three different chambers. So it's important to remember these three parts of the ovary, the presence of the ovules. For example, here they are in the center, they are attached to the placenta that provides nutrition, and finally placenta can form septa, okay, and that helps in the formation of chambers called as locules. Now locule concept is very important, especially when we study about fruits a little bit later, so you need to know this really well. Now let's go into the structure of the ovule very specifically, or the megasporangium. Okay, so the megasporangium implies it contains the megaspore. Okay, so the megaspore probably relates to something with regards to the female egg cell. Okay, it's not the female egg cell, but let's say in relation to. But let's study about the ovule or the megasporangium. Now, a typical megasporangium is what you can see over here. 70% of angiosperms have a megasporangium that looks this way, an ovule that looks in this particular form. Okay. Now, let's try to study some important parts of this particular ovule. The first important part of this ovule is, we said that the ovules are attached to the placenta and they draw nutrition from the placenta. So the part that helps it in attachment to the placenta is this elongated structure that you can see called as the funiculus. Okay, so this funiculus helps in attachment of the ovule to the placenta. But remember, we attach ourselves to the placenta because we want to draw nutrition. So you can see in the funiculus you have this dark strand over there, and the dark strand very clearly indicates vascular strands. So how you're going to pull in material. Okay, so xylem and phloem basically. So vascular strands are going to be present over here. Now, so this is how you're attached. Now coming to the actual ovule. The ovule has got two ends. One end is going to be called as the chaliceal end. Now for our figure, we look at it on top, we see it on top. And you have the micropyle end which seems to be below. But I want to draw your caution to this particular part. The typical angiosperm ovule that we are studying over here is seen in around 70% of your angiosperms, most of the angiosperms. It is called as an anatropous ovule. Okay, now this particular anatropous ovule is actually an inverted ovule, and since it is an inverted ovule, though we draw it in this particular form for simplicity's sake, and according to this figure, the chaliza seems to be on top and the micropyle seems to be below. If an anatropous ovule is actually an inverted ovule, the chaliza, technically speaking, is the base, and the micropyle, technically speaking, is the top portion. So we need to remember this part. It's very important for your NEET exam, and they tend to catch you on this because if you look at the typical angiosperm ovule, it looks like vice versa. So you need to remember, chaliza represents the base of the ovule, micropyle represents the top of the ovule. Okay, now let's focus a little bit on the micropyle. Now the micropyle is also called as a micropyle opening. It is an opening of something. Okay, so very clearly you can see that the micropyle seems to be covered with two layers. Very characteristically speaking, you can see two layers over here. Okay, on either side, presence of two layers. Now what are these two layers? These two layers are going to be called as integuments. Okay, and these integuments are nothing but protective coverings, and they're protecting certain important tissue on the inside. Later on, these integuments will help you to form the seed coats, but right now they're just forming some protective coverings. So the opening of the integuments is actually called as the micropyle opening. Now, the integuments basically protect two important entities in the center. The first is the nucellus. Now, the nucellus represents all the dotted structure that you can see within the integuments. Okay. Now, these nucellus cells are highly nutritive cells. They are diploid cells. Okay. So they're secreting a whole lot of nutrition that will be provided probably to the centermost part that you can see, the center part over there, called as the embryo sac. Okay. So the integuments protect the nucellus and the integuments protect the embryo sac. Now we will focus our attention to the embryo sac because that is the most important part of this particular ovule. Now the embryo sac technically contains three important cells, and all of these cells are haploid in nature. We will understand a little bit later in later videos why haploid in nature. But for right now, just remember all of these cells are haploid. The first cell that it has is the antipodals present at the chaliceal end, followed by the polar nuclei more or less in the center, and then at the micropyle ending, you have the egg apparatus containing one egg cell and two synergids. Now, to observe this a little further, to visualize this better, let's take this particular figure of only the embryo sac. So on top, you can visualize the presence of antipodals very clearly. They are three in number. Okay. Now, later on during fertilization events, you will find that the antipodals have absolutely no function. So if something does not have a function, after some time, these antipodals will degenerate. They have absolutely no purpose. Okay. Now let's go to the opposite end, that's the micropyle end or the micropilar end. Now here very clearly you can see the presence of the egg cell, again a haploid cell, and you can see the presence of two synergids. Now together, two synergids plus one egg cell make up what we call as the egg apparatus. Okay. Now the synergids are also haploid cells. Let us focus on the structure of this particular synergid. It has an important feature. Now the synergid seems to have these finger-like projections. Both of them have these finger-like projections called as the filiform apparatus. Now what is the importance of this particular filiform apparatus? Later, when the pollen grain touches the stigma and the pollen tube germinates through the style, the pollen tube will enter the ovary. But remember, the pollen tube contains the male gametes and the male gametes want to fuse with that egg cell present there. But the question is, how does the pollen tube find its way towards the egg cell? So this particular filiform apparatus, either one of them, okay, either synergid, not both, will allow the pollen tube to enter and will guide the pollen tube towards the egg cell using those finger-like projections. And as it's guiding, you will find that the filiform apparatus and the synergid will begin to degenerate. And in essence, by degenerating, it is making a way for the pollen, or rather for the male gametes to come in direct contact with the egg cell and therefore allow fusion. Okay, so that is very important on part of the synergids and the importance of having a filiform apparatus. Now, so here we see the filiform apparatus has pollen tube entry, guides pollen tube entry. Now in the center we have something called as polar nuclei. These are two in number. Now always remember that polar nuclei are haploid cells. However, these polar nuclei sometimes can fuse together to form something called as a central cell. Now, if they fuse together to form a central cell and the nuclei fuse, that particular nucleus is definitely diploid. So the central cell is diploid, and that diploid nucleus is going to be called as a secondary nucleus. Okay, it is a fused product, so therefore it is called as a secondary nucleus. Now, let's go through some important points with regards to the embryo sac. Okay, so the embryo sac, also called as the female gametophyte. So remember, gametophyte is something that houses the gamete, so it houses the egg cell as we have studied. Okay, now the embryo sac we know is made up of these three types of cells. Now let's count the number of cells present within them. You have three antipodals, you have two polar nuclei, you have three cells in the egg apparatus, that is one egg cell plus two synergids, which means technically speaking the embryo sac is 8-celled and 8-nucleated, provided that all these nuclei are going to be haploid. However, if a central cell is formed, as we can see here, sometimes the polar nuclei may fuse to form central cells. So if the central cell, which is a diploid cell having the secondary nucleus, is formed, the formula changes a little bit. Okay, you will have three antipodals, you will have one central cell instead of two polar nuclei, and you will have the presence of three cells in the egg apparatus, which means the embryo sac is no longer 8-celled, 8-nucleated, but it is 7-celled, 8-nucleated. Now, this particular concept of being 7-celled, 8-nucleated is very, very important for the NEET exam, and you need to understand how it comes into picture. Okay, and you need to remember that the central cell nuclei, being the secondary nucleus, is definitely going to be a diploid nucleus. That is important for you. So let us go through a couple of MCQs to familiarize ourselves with whatever we have studied. The first, a mature embryo sac represents first the megaspore, female gametophyte, the sporophyte, or the megaspore mother cell. Now, very clearly, what is it representing? The embryo sac contains the female gamete, okay, the egg cell. So definitely it has to be the female gametophyte. The next, number of cells in a typical mature embryo sac are. So how many cells would you find? So a mature embryo sac would imply that you're moving into the 7-cell stage. Okay, so the answer is option A, 7. The third MCQ. If the stalk contains 24 chromosomes and the scion contains 12 chromosomes, how many are present in the secondary nucleus, the egg cell, and the root hair cell? Now, we are using concepts of the first chapter in the second here. Now, if you remember, the stalk implies we are talking about the root system, and the scion implies we are talking about the shoot system. Okay, so very clearly we know that the root hair belongs to the stalk, so definitely 24 chromosomes. And by looking at that, we clearly know that our options are either A, C, or D. So we can eliminate B out of this. Okay. Now, the scion implies we are talking about the shoot system containing 12 chromosomes. Here you have the secondary nucleus and the egg cell. Between these two, we know that the secondary nucleus is going to be diploid in nature, okay, whereas the egg cell is going to be haploid in nature. So secondary nucleus will be 12 chromosomes diploid, and egg cell will be half of that, and it will be 6 chromosomes. Okay, so therefore our answer is option D, 12 secondary nucleus, 6 egg cell, and 12 for the root hair cell. So I thank you students for attending today's video. I hope you have understood the pre-fertilization structure of the gynoecium. Okay, and in the next video we will be studying about megasporogenesis in depth. Kindly go through the different material provided under EduRev Infinity. You will have a whole lot of question papers, other videos, and you will have a lot of material to read through that will help you with your NEET exam. I'm sure by going through all of this, you will increase your nationwide ranking. Thank you, students.